In this video, we will discuss about this congenital disorder of glycosylation. So, in this kind of diseases, what will happen is that there is a problem in the glycosylation, and there are two major glycosylation that occurs in the mammalian cells, that is end-linked glycosylation and the O-linked glycosylation. And some of the drugs that are being used to treat or diagnose this kind of disorders are under clinical trials. So we will discuss those drugs also, how they work towards diagnosing these diseases. So we will start with this end-linked glycosylation disorder. So in the case of end-linked glycosylation disorder, this PMM2 enzyme is not working properly, and because of that, it will lead to the improper glycosylation. And one of the uh, problem that caused because of this improper glycosylation is that this calcium channel will get hypoglycosylated in a way that it will allow more calcium ions to go inside the cells, and that will create problems in the cells. So one of the drugs that are being introduced is this man. So purpose of this drugs is to accelerate the normal glycosylation process. That is, from the mannose it will convert it into this mannose 6 phosphate, then to this mannose 1 phosphate, and thereafter this GDP mannose, and the glycosylation pathways goes on. So this drugs, we can say that, trying to compensate the need of this PMM2. The other drugs, that is rested CHCA, its function is to produce more PMM2 enzymes, so that the low efficiency of PMM2 enzyme will be compensated by the high production of this PMM2. So how it does is it will reduce the side product formations in the process. For example, this glucose to sorbitol formation will be inhibited, and that will cause more production of this PMM2. And more production of this PMM2 will attract more chaperones such as HSP70 and HSP90, and the protein folding will be improved, or we can say that in the final outcome we will have more PMM2 that is properly folded. So this is the second kind of drugs. This is the second drugs that have been tested in order to diagnose this disease. And the third drug is that this cholesterol. So in the case of cholesterol, what will happen is that instead of degrading this instead of degrading this non-native folded PMM2 to producible degradation, what it will does is it will again send back this PMM2, non-native PMM2 structures towards the chaperones such as HSP70 and 90, and these chaperones will again attempt to uh, properly fold these PMM2 enzymes so that they can function necessarily. And the last drug that has been mentioned in this figure is about this acetazolamide. So as I have discussed, that one of the problem that occurs because of this PMM2 enzymes is that hypoglycosylation of this calcium channel. So this acetazolamide drug uh, helps in uh, helps in reducing those calcium channel pores by binding to the calcium channels. The clinical trials results has been found beneficial in terms of the intracellular results. However, the quality of patients in this case has not been improved much. So further studies are going on in this kind of drugs, in these drugs. Now let us quickly understand this O-linked glycosylation disorder. So in this O-linked glycosylation disorders, some of the problems that cause is that happen is, or disruption in this O-linked glycosylation will cause increase in these BMP receptors, and that will lead to the high amount of activin and other BMP ligand in the extracellular matrix, and this is usually occurs through this R206H mutations in this ALK2 gene, that leads to the overactivation and phosphorylation of this SMAD pathways, such as SMAD158 pathways. and that will ultimately lead to the high BMP receptors formations. So one of the drugs that are used to target this is this palavorotine, and this has been tested in the mouse model, and the positive results of this mouse model has led to this approach to go into the clinical phase trial. So what they have found out is that this drug, this palavorotine, will bind to this RAR gamma, and that will increase the degradations of this SMAD 158, phosphorylated SMAD 158. So instead of moving this SMAD 158 and 4 towards this nucleus and upregulates the genes that helps in this high BMP receptors formations, this SMAD pathways will be directed towards proteasomal degradations, and so the upregulations will not occur, or the epigenetic changes will not occur, and the BMP receptors will be reduced whenever this drugs is used, that is palavorotine. So this is how they trying to regain the healthy cellular functions in the case of all in glycosylation disorders, where these kind of mutations are found, such as mutations in ALK2, where R206 is mutated with H. So, so these are some of the drugs that are being currently under that are been studied under clinical phase trials. However, to the best of my knowledge, these drugs so far has not been approved for the commercial use. I don't have the recent status of these drugs, but the information that are presented in the paper mention that these are still under studies. These drugs are still under studies. So, if you need to understand more about this congenital disorder of glycosylation and what are the mechanism of this different drugs in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.